So the structure I'll use for today is to start with kind of an outline, kind of a big picture, talk about what it is that we want in a drug, what's the goal, and then talk about the pathway to get there, the players, who's involved, and some of the challenges in getting there. But then to illustrate this in more sort of detail, I'd like to use an example of one disease and one drug, and that will be multiple sclerosis and the drug Natalies Mag. And then we'll wrap up by considering some innovations in the field, and one that I think is really exciting called the tissue chip, or tissue on chip, which can hopefully sort of expedite or improve this drug discovery process. Okay, so let's start. What is a drug? Now this can mean different things in different social contexts. So for today, we will think of this as a compound or product that cures or alleviates a disease. And we've heard about a number of these in the other talks, right? We heard about antibiotics to treat bacterial infections. We heard about chemotherapeutic drugs for the treatment of cancer. We heard about antidepressants, right, in the psychiatric disease talk. And these can all be categorized as small molecule drugs because they are fairly small, at least in comparison to some other drugs, like proteins. So we also heard about insulin, which is around 100 times larger than penicillin, for example. Insulin is used to treat patients with diabetes. And another protein drug is an antibody. This is around 600 times larger than an antibiotic. An antibody is a protein that actually all of our immune systems can make, but scientists can also engineer these to sort of target or bind to, interact with whatever molecule or protein that they want. So they can end up being a very powerful drug as well. Okay, so those are some examples. Now let's say we want to imagine a new ideal drug, and I'm going to ask you, what do you think is the most important feature, or what are the few most important features that you would like in this new drug that you're going to take? Who can tell me what's important to them? Safety? Great, I totally agree. I hope something that I take is not going to hurt me, right? What else would you like?
Effectiveness.
Effectiveness, yes. Why would I take something unless it's actually going to help me? Is there anything else you can think of that might be important?
Targeted.
Pardon? Targeted, sure, that it's very specific in its effect.
Affordable.
Affordable, yes. Safety I agree with, and affordable I totally agree with, right? If there's this amazing drug but it's like $1 million, it might as well not exist to me. That's totally out of my reach. Unless I have a really great insurance policy that can pay for it, and then it's great, then it's affordable again. So I agree. I think efficacy, that it works, the safety, and affordability are really important. One factor that we don't always have the luxury of considering, but it's certainly still important, would be convenience, right? So is this a pill I take or an injection I have to give? Or maybe it's a patch that I wear instead of taking 5 pills a day. And certainly when a drug company is thinking about a product that they can make that someone like us might want to buy, this is a factor that they would consider that would be important for them. Okay, so this is our goal then, what we want to achieve, and what's the path to get there? Well, it's always going to start with sort of a problem. Not that any of us are a problem, but it's going to start with sort of a disease or some ailment in patients. First, this needs to be recognized, identified, before we can make any progress. And then to come up with this ideal drug, we're going to need to go through a few phases of research. And the first one we'll call preclinical research. So this is done by scientists at research laboratories, at research universities like Harvard, Tufts, BU, all across the country and the world. And the goal here is to understand how the disease works and try to come up with a potential drug that could affect that disease state. So this can be done sort of in a test tube, looking at just chemicals work. It can be done using samples of and very importantly, it also is done using animal models. And these models can be very useful to sort of mimic or model how a disease works in humans. And very importantly, they then provide a way to test a potential drug. See if that drug that we think might be involved in fixing a certain pathway, whether it can actually affect a disease outcome. So after this phase of research, sort of in what we call the preclinical phase, the next stage is what we'll call clinical research, and this involves testing in humans, testing that drug in humans. And previously we had these scientists and sort of research institutions involved. Now there's a couple more key players. The first one is what's called here a drug sponsor, or we could think of as a pharmaceutical company or a biotech company. Somebody with a lot of money, because clinical trials are very expensive. So you really need a group like this involved to sort of push the drug through the next phase of testing. But they are going to be working actually very closely with the Food and Drug Administration, the FDA, which is the regulatory body in the US. So the FDA sets all sorts of guidelines to make sure, like you want to run a clinical trial, you want to test this new compound in people, well, you better make sure that you've done your due diligence, that you really think it's safe, you really think it's going to work. And when you design that clinical trial, you want to make sure you're being cautious, that you're not going to kill a bunch of people or cause them a lot of harm in the way. So the FDA sets up all these guidelines for what the clinical trial needs to, what it needs to demonstrate in order to convince them that this is a drug that meets these goals. Now, if a drug is so lucky as to be approved by the FDA, this is the point where we then have a chance to usually interact with it, right? Now it can be prescribed by a doctor, or we can buy it over the counter, and it has a chance to affect us. Okay, so here's the pathway. Now, what are the challenges in this process? I mean, obviously we don't have like millions of new drugs arriving on the market every day, so there must be some kind of hang-ups. The first one is that it tends to be a very lengthy process. Actually, a study was just completed last year, which found that on average, it takes around 14 years to go from sort of identifying like what it is you want a drug to do, identifying a target, to actually getting FDA approval. That's a really long time, especially if I'm someone who maybe has a progressive disease, it's getting worse every year. That's a long time to wait. The second challenge is that this process is very risky. And the same study found that of the drugs that actually enter clinical testing, enter testing in humans, around 85% of them don't get approved. And actually, some of the sort of calculations or estimates on this number are even higher, like 95%. Now, if we do a little math and we think about, all these really long projects, most of which don't end up revealing a drug product that works, we come up with a really big number for, on average, then how much it costs to come up with one successful drug. And this is something that we kind of end up experiencing then, right? Our drugs are not free, they cost us money. So why do these challenges exist? Well, I'd like to use the example to sort of go into these in more detail and understand why that is. First, I'll pause for questions. Does anyone have questions so far about this pathway or the players? Okay, good. Okay, so the example I'm going to use is multiple sclerosis. This is a disease that affects around 90 individuals per 100,000 in the US. Possibly someone with MS. I actually definitely do. And this is a disease that causes damage in the brain to this myelin sheath, this insulation that surrounds axons. So the way that nerve cells in the brain communicate is by sending an electrical signal. And this myelin acts as kind of an insulation, just like plastic on a wire, to allow that signal to go a long distance. And when this is damaged, that signal breaks down. So now the brain tries to send a message to, let's say, the legs. Oh, we want to go for a walk, or we want to wave the arms. When that message breaks down, it doesn't work as well. They don't get the message, and the muscles don't respond. So what a patient experiences then is sort of this weakness. And although it's an area of active research, the current understanding of why this happens, or how this happens, is that it's actually cells of the immune system that cause this damage, which is kind of weird, right? Usually our immune system protects us. It fights bacteria, it fights viruses. In this case, it's actually harming our own body. So this drug that I want to use as an example, called Natalies Mag, actually prevents immune cells from getting into the brain, thereby preventing damage from occurring, preventing progression of the disease. Okay, so let's go through sort of the history of the development of this drug, and keep in mind sort of who are the players that were involved, what are the steps to get ultimately to this drug that was approved, and what are some of the challenges that were encountered. So MS was first identified as a disease in 1868, kind of a long time ago. And it wasn't until around 100 years later, in the 1960s, that the first animal model of MS was developed. And this was done in rats and in mice, and is still used today. And as I kind of mentioned before, sort of the value of animal models, this was a really critical sort of development in this preclinical research phase to be able to investigate how this disease might work. What's going wrong? What's different in this disease state versus a healthy state? And how can we try to affect that? Now, scientists who are studying this animal model, as well as some samples from humans, discovered by the 1970s that this disease seemed to be caused by the immune system. Now, totally separate from this study of MS, other scientists were interested in how the immune system works. And specifically, they wondered, how do cells from the immune system go from circulating through the body, through the blood, and get to one particular spot where there's an infection? So say I have a cut on my hand, there's some bacteria there. How does an immune cell decide to leave the blood and go right there and fight that off? And what they found was that there were certain proteins on the surface of immune cells that allowed them to go to specific tissues. Now in the 1990s, scientists who were studying MS said, hey, we have this disease here that we think is caused by immune cells going to the brain and causing damage. These other guys found a way that immune cells get to certain tissues. Maybe there's a similar mechanism that allows immune cells to get to the brain, and that could be an area that we could target with a drug. So here's kind of what they were thinking. They thought, how do immune cells enter this inflamed brain tissue? Maybe there's an interaction between some unknown, so far unknown molecule on an immune cell that's going to interact with a receptor on a blood vessel that goes through the brain, and that this interaction will allow a cell to enter and cause damage. But if they could come up with some sort of compound, some sort of drug that would block this interaction, then the cell could go by, never enters the brain, never causes damage. So to approach this question, they considered all these receptors that had been identified by other scientists and said, maybe one of them is important in this case. And so they tested them. They tested them one at a time. And they used different compounds to block this interaction. They actually didn't really have to invent these compounds. They used antibodies, which had been developed for the research purposes. And they were able to block each of these interactions and see if that had an effect on disease. And in fact, one of them was very important. All the other ones weren't, but there was one that they identified that was critical for these mice to get the MS disease. This was very exciting. And so they took the next step to take this into clinical trials. So some of the scientists who were doing this work, the group that actually first published was at Stanford University, and they partnered next with a pharmaceutical company to alter the drug a little bit. Previously it worked in mice. They needed to make a version that would work in humans. And then started to take this into the clinical trial phase. Now, before I tell you the story of Natalies Mag in clinical trials, I want to give you a little bit of background on how clinical trials work. So there's a few features for how clinical trials are set up to hopefully allow these scientists to interpret the data best. So like setting up any experiment or test where you want to know an answer, we want to make sure that if we're testing an agent like a drug and we're looking for an effect, that if we see that effect, it's actually because of the drug and not because of some other factor that we forgot to think about. So there's a few ways to try to help this happen. One of them is called randomization. So let's say we have some patients in this trial. Some of them are from Seattle, some are from Boston, some are from Orlando. Now when they go into this trial, we're not going to put everyone from Orlando in the same group, right? Because, I don't know, maybe sunshine has something to do with this. So we're going to split them up randomly. The second thing we can do is use a placebo. Probably you've heard of a placebo, right? This is going to be like a sugar pill. Maybe if it's an injection, it's just injecting saline. And the idea is that everybody who's in this trial, both group A and group B, they get the same treatment, except one gets the drug. Okay, everybody goes to the doctor once a week, they get that injection. The only difference, as far as we can control, is that one of them happens to get the drug. The third feature is that this is blinded. And this gets at that concept, maybe you've heard of the placebo effect. The idea you're taking a pill, you're like, oh yeah, I think I feel better. So the idea here is that the patients don't actually know if they're getting the drug or they're just getting the placebo. And actually the physicians, the doctors who evaluate their disease progression, they also don't know. So everyone's trying to be sort of not biased, try to eliminate that bias in the process. And the fourth sort of component, which I'll just touch on, would be the study size. So more people, more money. But more people, you can have more confidence in your data, right? 1 person in 10 has an effect from the drug, let's say. I don't know, maybe that's just due to chance. Maybe they were kind of a funky person. But if you actually have like 10 or 12 people out of 100 that show that effect, now you have a little more confidence that it's actually because of the drug, not just by chance. Okay, so these are some features of the clinical trial. Now when they're actually run in phases. So we said earlier those features that we thought were important for a drug: safety, efficacy, affordability. If you were going to run a clinical trial, what would be the first thing that you test? Safety or efficacy? Safety, yeah. I agree. Sometimes actually they're done at the same time. So oftentimes in trials for drugs for cancer, they'll test sort of efficacy and safety at the same time. But generally you're going to start by just testing safety. Make sure it doesn't cause any harm. And unfortunately, there are cases where it actually does cause a lot of harm. So we're going to start with just a small group, healthy participants, in case anything does go wrong. Hopefully they can recover. And if a drug does pass this safety phase, Then you can go on to testing efficacy. And we'll start with, again, kind of a smallish group. Now we're going to test it in patients, see if it works. And if it shows promise at this phase, then you can move on to larger trials. Thousands of patients, multiple sites, independently run to really try to get an idea if there's data that this drug works. Now, unfortunately, drugs don't really pass all of these tests, and actually these failures happen at each sort of stage of this process. So in the evaluation of safety, around 33% of the drugs that enter phase 1 trials don't make it past. They're deemed not safe or not safe enough to make it to the next stage. And even of those drugs that are found to be safe and go on to be tested for how well they work, 42% of those are found to be not effective, or again, not effective enough to warrant going on to the next stage. And even those ones that did show a pretty good effect and they went on to phase 3, well, around 60% of those are suspended as well. And when we look at the reasons for why those drugs in phase 3 don't make it to FDA approval, it's those same reasons that we considered important at the beginning, right? Primarily, they don't work well enough. Maybe they're not safe, although most of the unsafe drugs were kind of screened out at the beginning. Or there's a commercial reason. Maybe the company ran out of money. They can't fund this project anymore. Maybe they realized it was more expensive than they expected. There's not as many people who are going to buy it as they originally thought. It just doesn't make financial sense for them at that point. So why are there so many failures? Why is it that this safety and efficacy are so hard to predict in clinical trials, even when a drug made it through that preclinical testing? Why is it that when we start with, say, 100 drugs that go into trials, we keep losing more and more so that at the end, only around 15% make it through? Well, the primary reasons are listed here. First of all, research models of disease, laboratory animals, test tubes, are not the same as humans, as much as we might like it. There's just biology that's different. And if we did try to study human biology more specifically, unfortunately, the samples or materials available to us are pretty limited, right? We can easily take a blood sample from someone. That doesn't necessarily tell us what's happening in the brain or in the liver. And taking a sample of brain, I'm not going to be the first one to volunteer for that, right? And even if you're taking a sample of a cancer or tumor, it's only going to be a little bit. It's going to be hard to learn very much from that little tiny sample. Another factor that kind of becomes more important as time goes on is that each new drug that goes through this process needs to be somehow better than a current drug. So the FDA requires that a new drug is going to be either more effective or more safe than something that's currently on the market in order to get their approval. These are really high standards. So I'll pause here for questions before I go into our example and see how Natalies Mag did in its clinical trials. Any questions right now about the preclinical research on Natalies Mag or how clinical trials are set up?
Is there a strict protocol for starting a clinical trial? Is there a requirement list of whether that has been done on animals, what kind of animals, on tissues, what kind of tissues, and things? Are they the same internationally?
Good question. So the question is, are there strict standards for the requirements of a drug in preclinical testing before it's tested in humans, and are they the same internationally? So there are strict standards, but they are not the same internationally. So in the US, it's the FDA that sets those rules, and actually animal testing is required. It needs to be shown to be safe and effective in animals before you can ever try to start something.
Which animals?
Sure. So that could depend on the disease and the models that are available. So oftentimes there are models in mice. Sometimes there's also models in chimpanzees and monkeys. And so depending on whether that model exists for a particular disease, that might be required as well. So the idea is to sort of do as much as possible to find out if it's going to work before testing it in a human, since that's such a risky endeavor. And different countries have different regulatory bodies that set those rules. So the UK has its own, or Europe has its own, but they would probably be pretty similar standards. Any other questions about this process so far? Yeah?
Do you see any noticeable trends that decrease the 14-year cycle or increase the lower the percentage or increase the percentage of efficacy?
Yeah, like the success rate and stuff like that. So I think actually in the report that I saw, it was a little bleak. I think that the time to discovery or for a new drug and that success rate had kind of dropped. But that was partially due to the fact that, like I mentioned, like each new drug needs to be better. There's kind of like a higher standard each time for each new drug. But as I'll get to at the end, there are a number of innovations and sort of goals in progress to improve the discovery process. Okay, I think I'll move into my example, but we can take more questions later. Okay, so let's see how Natalies Mag did. Well, it had these really great preclinical results, and actually it had very good clinical testing as well. So went through this safety and this first test for efficacy between 1997 and 2003. And in 2003, 2 independent phase 3 trials were initiated. And this was planned to be, I think, a 2-year study, but actually just 1 year into the study, the FDA looked at the evidence on how well it worked and how safe it was, and they said, you know what, this looks really good. And they granted approval. So they actually expedited the approval of this drug based on the evidence that they saw for how well it worked. But even with this expedited process, we can still see from that time of sort of target discovery, of identification of this particular protein that was important for cells getting into the brain. That was in 1992. Approval in 2004, that's still 12 years. And that was taken up with modifying the drug from its original form to one that would work in humans and going through the many phases of the clinical trials. Now, unfortunately, that wasn't really the end of the story for Natalies Mag, because just 3 months after it was approved, there were 2 reported cases of death in their clinical trials. And so it was the company that made it actually voluntarily stopped making it. They stopped marketing it. They said everyone should stop taking it because, there have been 2 deaths and we don't really know why. But it turned out that when they investigated, these deaths were due to inflammation in the brain caused by a virus called the JC virus. This is a virus that actually most of us have, 50 to 90% of us have it, but don't panic. For most of us with good immune systems, it's not a problem. We keep the virus under control. It tends to be a problem for patients with compromised immune systems like HIV, or if you've had an organ transplant and are taking immune suppressants, then the virus can kind of get out of control. And it can cause this disease called PML, progressive multifocal leukoencephalopathy, which basically means worsening, spreading brain inflammation. Really not a good thing. So at first it was really concerning that this happened, and scientists tried to investigate what was the link. Why was this happening to patients taking Natalies Mag? And what they found was the following. Oops. So we know that Natalies Mag prevents immune cells from getting into the brain, which is really great for treating MS, right? Because those immune cells are the ones causing damage. But in the case of a person who has the JC virus, if this virus gets into the brain, in a healthy person, immune cells can go in the brain and take care of that virus, keep it from spreading too much. In a person taking Natalies Mag, now those immune cells can't get in. Now that virus can sort of expand out of control and lead to extensive damage in the brain, which, as we saw, turns out to be fatal. Now, this is a really big risk. Maybe we would think that a drug that has this potential to kill someone should not be approved. But actually the FDA, again, evaluated the evidence. They also listened to patients. There were a number of patients who felt this drug worked so well for them. It really stopped the progression of their disease. And the FDA felt that because they could identify the JC virus as this key risk factor, they felt comfortable reapproving it. So just 1 year later, they decided to reapprove Natalies Mag, but now there were more restrictions for its use. Everyone needed to be tested to see if they had this JC virus first. And if they did have it, it was recommended that they not take this disease. And everyone who not take this drug. And everyone who started on this drug needed to be continually monitored for the virus. They were sort of entered in this database to monitor them, to look for side effects, and really try to evaluate what was the cause for, or sort of identify additional risk factors for why some people had this complication. I think this highlights a really important point about the decisions the FDA has to make, right? They're kind of tough. They want to make safe drugs available to people. They want to ease suffering, but they also have this responsibility to make sure that they're not putting something out there that has the potential to cause a lot of harm. Okay, so I'll summarize a few key points from this story that I think are really key in this example. So the first one is that we saw the discoveries in a field, it wasn't a study of disease, it was a study of the immune system, how the immune system works, sort of biology. This was actually key to the development of this particular drug. And I think this is a really common feature in discoveries in medicine, that it's something sort of in a different field, almost unrelated, that ends up leading to a really important discovery or treatment. The second thing, we talked about how there's this high failure rate in going from the preclinical studies to the clinical studies, and that humans are different from mice. And we saw a perfect example of that, right, in this complication with the JC virus. No one would have thought that this JC virus would end up being a problem for this drug. Mice don't have this JC virus. It would be pretty much impossible to predict that this could happen. The third thing, which I guess I just mentioned, is that the FDA has this really complicated task, or sort of tricky task, of, again, evaluating evidence, deciding if something seems safe, wanting to make drugs available, and ultimately taking the responsibility for deciding what should be available to people like us, who don't always have the opportunity to evaluate all the evidence. Okay, so we talked about those sort of challenges along the way, why it's hard to come up with a really great drug that works and meets our criteria of safety and efficacy. So what are some ways to address the challenges? Okay, so wait a minute. Okay, yeah, that's fine. Okay, so one method is called repurposing of approved drugs. So actually, now that a number of drugs have been through this process, they're found to be safe, they're used for particular treatment of particular disease, now they're actually available for scientists and doctors to think, to try to apply them to another disease. So actually, Natalies Mag is used for multiple sclerosis, but it's also used to treat Crohn's disease. And actually, every drug that goes through this clinical trial process is only tested for its use for one application. So there were a number of clinical trials for testing Natalies Mag for multiple sclerosis. There were separate clinical trials to test this drug for treatment of Crohn's disease. And actually another really common thing lately is that there are drugs to treat cancers of the immune system, like lymphomas and leukemias, and those turn out to be really great drugs for treatment of other disorders of the immune system, like rheumatoid arthritis and actually even multiple sclerosis. Another way to try to address these challenges is to improve our laboratory models of disease. So one way to do this would be to sort of better understand the current models we have, know what their limitations are, know that they can't really predict how everything is going to work in a human, and to really try to come up with new models that can provide better examples of humans. And the third thing is to come up with new technology that lets us do more with less. And this really gets at the opportunity to try to learn from human samples. So there's a number of technologies now that can take just a tiny clinical sample and actually extract so much more information from it, because it works with like really tiny volumes. You can work with single cells, and you can get a lot of information now from a very tiny sample, whereas in the past we could only get a small amount of information. And one particular example, which I think is really exciting to improve this process, is this thing called a tissue chip, which looks like this. It's a piece of plastic around the size of like a quarter or a key, and the idea is that you can put some human cells in little like wells here, little areas, grow these human cells to mimic a human tissue. So you could take cells from, skin or heart or liver or kidney and grow them here, and then be able to test how drugs affect these particular cells. And the idea is that this would be kind of an intermediate step between that preclinical testing, which is primarily in test tubes and animal models, and before you actually go to testing in humans, use this model that uses just a little bit of human cells and evaluate toxicity of drugs at this phase. So if we look here, with this piece of plastic has little sort of engraved channels on it, and if we zoom in, we see this series of diamonds. If we zoom in 2 times more, we see that what's here, actually this blob is tumor cells that are growing, in this particular one. They're growing tumor cells, and these lines are little blood vessels. And there's actually a whole circulatory system set up on this chip to kind of mimic how our own circulatory system works. It flows in oxygen, takes away waste, and this is now a place where multiple drugs could be injected kind of in parallel, and you could assess, say, does it kill the tumor cell but not kill blood vessels? Or does it kill the tumor cell but not kill kidney cells? I think this is a really exciting area that the National Institute of Health, NIH, has put a lot of money into this program. It started in 2007, and there's multiple sites across the country already sort of developing particular tissue chips for particular tissues. I think it's something really exciting. Okay, so to summarize, what we've talked about in this session then is sort of this goal we had of the ideal safe and effective drug and how we got there. It started with observation and understanding of disease. These are 2 phases of research. The preclinical phase, which involves testing in test tubes in animals and involves scientists like those who hosted this event for you today, taking place at universities, and also this phase of clinical research, which now brings in pharmaceutical companies, biotech companies, working with the FDA to hopefully come up with some drugs that meet these really high criteria for us. Okay, so with that, thank you for your attention. Thank you for attending today. Also our sponsors and organizers, and I'm happy to take questions.
The last you mentioned could be an intermediate between like preclinical and clinical testing. Could it also be used for drug screening, like to identify targets or leads?
To identify targets or leads. Yeah, I suppose so. I guess I usually think of target identification as a little bit more molecular, like saying what is the protein that you want to interrupt. But I think, yes, I think you could. You could still say, here we have a potential drug that or I would say a compound that we know interferes with this pathway. And that could also be used in this phase to see if it has a certain outcome. You would just need to have a planned readout, like here's the effect we want to see, and then do we see it or not? So yeah, absolutely could be used for that purpose.
How about orphan diseases? How do they get developed, the rare diseases?
Yeah, so we kind of touched on how this is also a commercial endeavor, right? Not just doing good for the world. So actually, so orphan diseases are not always neglected. Sometimes they are funded by a group like the Bill and Melinda Gates Foundation. Maybe it's a charity that can provide money for a company to do research on that. There's also oftentimes money raised by sort of patient advocacy groups. So I know there's groups for like patients with ALS, right? We saw this water bucket challenge. They're raising money to try to support research for this disease. Also for multiple sclerosis, for breast cancer. It's not really an orphan disease, but we certainly hear a lot about sort of patient advocacy to raise money for this. So there's money available for those as well. And I think a lot of the research that starts happens at academic institutions that don't always have that commercial bias, right? They're just interested in kind of generating knowledge, understanding how things work. And that step for commercialization definitely depends though on where the money comes from.
I think for the tissue chips in particular, I think you're talking about Charlton Hamilton's new company. I think they just got a $50 million endorsement.
Okay, that's really cool. Yeah, actually, tissue chips are also sponsored by the government for research at universities in the US. But yeah, I'm sure there's also companies that are involved in making them as well.
I have a comment about your affordability.
Okay.
I think affordability is very different from cost effectiveness, and it depends very much on the budget, on who's paying for it and what the budgets are. And I think a good example of that is the new hepatitis drug that came out, which is much more cost effective than anything else, but because it cures things very quickly, but because the paying agencies have to pay for it up front, it's not in their budgets, so they refuse. They're waiting for competitive products to come out and drive down the price, and are not making it available as much as they otherwise could in the lowest FDA.
Yeah, so the comment is about, if you have multiple drugs that are available, maybe they both kind of work, maybe one is better. How can you decide if one is cost effective, especially if it's an insurance company that's paying for it? Actually, there was one study on Natalies Mag in a European nation where there is socialized healthcare. So actually, it's the government paying for it. And they actually did a study. So Natalies Mag is more expensive than the other sort of currently available treatments for MS. But they did a study. And it sounds like really, unempathetic or crass or something. But they evaluated like how expensive each drug was, and ultimately sort of the total cost effectiveness. And when they factored in, how well it reduced disease progression, how that reduced the number of hospital visits, of additional drugs needed to kind of contain the disease. Actually, even though it was more expensive than the other main treatment, it ultimately costs less to use it. And so they actually decided to support the use of that one. And definitely that's always a factor for, when we're not paying out of pocket, even if we are paying, certainly the cost ends up mattering. Yeah.
The drug companies are doing the testing, right? Supporting the testing. Is it how often? And the FDA just takes their word for all their evidence, right? That they are showing that it's safe and effective.
Yeah. So the company would provide that data to them, and yes, absolutely, there is an expectation that they are being truthful. There are huge. All the science is kind of assuming people are truthful, and if you're found to be not truthful, there are huge repercussions, right? Because then no one is going to trust you in the future, pretty much. So it's actually kind of a lot on the line. But absolutely, there is, some trust involved there.
Great.
Great. Well, thank you, Shannon, so much for ending our day on a